book has that merged in with the part that's on cell anatomy out of chapter 4. We'll be looking at that. I also have a question set for you to work on with other people here on that same thing. So the lecture is not going to take all the time we have between now and 9:45. Also planning for us to watch a short film, in that case dealing with lactose tolerance. As you know, some people are able to drink milk as an adult and they don't have digestive issues. Other people can't. And so it'll be looking at that difference. I have a question for you right now to think about. Somebody there is a mutant. Either the people who can drink milk as an adult or else the people who can't. Which ones are the mutants?
We cannot see your screen.
Right now, I am not sharing anything.
The people who can drink it are mutants?
Possibly. Yeah, possibly. Or maybe the people who can't. I mean, we talk about lactose intolerance, like, what is this, terrible genetic condition. Well, maybe it is, maybe it isn't. We'll be seeing about that a little bit later here this morning. Okay, so that's something to think about. Otherwise, I am going to take us through the rest of what we had from the chapter 4. And now I will be sharing the screen. Right, so membranes and transport. Right here is an example of the kind of thing we'll be looking at. You have here a protein, and you can see it's part of the membrane. The membrane has all of the phospholipids that are shown with the two layers, a little, or like near the middle, bit toward the top middle. So you have all those phospholipids, and they're providing the basic structure of the membrane. But when we look at real membranes, they always have a lot of proteins in them too. Here's an example of one of the proteins. This is embedded in the membrane. It's resonant in the membrane, and this one is implicated in disease. Look at the name of this protein. Does that look like it might be involved with a disease?
Yeah.
Like what?
Cystic fibrosis.
Exactly, which is a very serious disorder. People with cystic fibrosis don't live a normal lifespan. They have a real struggle even to make it to, basically about a lifespan of 35 or 40. It's a pretty bad disease. It's inherited, and it involves this protein. The thing is, everybody is supposed to have this protein. This is a protein we have a gene for. It transports chloride ions, Cl- ions, across membranes. Those are the kind of red circles that we see moving up with this big red arrow, right through a pore. And this protein provides a passageway for chloride ions to move from one side of the membrane to the other. This is something everyone is supposed to have. So it might seem strange that a healthy person has this thing called cystic fibrosis, transmembrane conductance regulator protein. That's a mouthful. But it's something where when this is faulty, people have the disease. Everybody with normal health has a normal version of this. And so we give it that name to just indicate this is where the disease is connected to if you're unlucky and you inherit a corrupted version of the gene from making this protein. But that's an example of membrane transport and the proteins in membranes that make it possible. Otherwise, if you don't have this protein there, these chloride ions can't get from side A to side B. And we will see later in the quarter, in another day or so, we will see about what happens with this disease. As it is, membranes will have this lipid bilayer we've looked at, mostly made out of the phospholipids, also cholesterol in animals, and proteins. And just at a very crude guesstimate, proteins or the membranes are going to be about half the lipids and half proteins by weight. That ratio varies a lot. But you can think of the membrane in some ways being just a structure to attach proteins to. I mean, it does work in its own right, the membrane does. It provides a boundary between different compartments of the cell, and that boundary controls what moves from one place to the other. Most substances are not able to pass through the lipid layer unless there's a protein there to move them. And so that means that if you have substances in one compartment of a cell, they will stay there unless the cell decides to move them somewhere else. That applies to most kinds of substances, like those chloride ions. And so it allows the cell to have different sections doing different jobs. So the membrane, the lipid portion of it, the water repelling portion, keeps most things either on one side or the other of the membrane. Then the proteins allow the cell to move things from one compartment to the other, but it does it under control instead of things just randomly going in different places. So we have this arrangement where we've got the double layer of the lipids, double layer of the phospholipids, and they separate a fluid space that is mostly water from a different fluid space. This could be an internal organelle inside the cell, or it might actually be the whole cell surrounded by, say, water. So depending on what scale we want to look at this, but the membrane separates these places from each other. Questions for me on this so far?
So the proteins that are embedded in the lipids don't move, but there are also free floating proteins as well that can move around.
There are free floating proteins floating in the cytoplasm. We have other proteins that are embedded in membranes. And if the protein is embedded in a membrane, it stays there.
Okay.
Okay. So the membranes, they have two surfaces. They're contacting two different water-based media. One of them could be the cytoplasm in the cell. The other surface could be touching fluid inside of a compartment like a mitochondrion, or it might actually be fluid outside the cell. But any way you look at it, the surfaces of the membrane are water attracting. They have those polar head groups as part of the phospholipids. So those surfaces want to swing around and be in contact with water and other polar things that would be dissolved in the water. Then you have the middle of the membrane, and that is a water repelling region that hides away from the water that is in the cell. And polar substances that would be dissolved in the water aren't able to pass through that water repelling region. Not very much, and many of them can't at all. Things like salts are totally unable to pass through the water repelling region of the membrane, except if there's a protein there that moves them. Now, the parts of the membrane are fluid. They flow from place to place, except if they are anchored. Some of the proteins are going to be attached to the cytoskeleton and they're not going anywhere, but others are free to move, as we will see here. So this is a demonstration of a hideous experiment that some mad scientist did. This mad scientist made a hybrid of a human and a mouse. Imagine that. Not really a crazy mad scientist. It sounds like it when you describe it like that. What they did, and this is back around 1970, the scientists took a human cell and a mouse cell and they fused them together. So they made a human-mouse hybrid. Oh my God. Well, this was to study the movement of the proteins that are in the cell membrane, the plasma membrane. What they did was they created a special line of human cells where the proteins in the membrane had a red fluorescent dye. That means if you expose it to, say, UV light, you'll get these little red dots lighting up. So the cell would have these little red dots all over the surface from the proteins that are in the membrane. They did the same thing with a mouse cell, except there it glowed with green dots in its membrane proteins. Then, with a little bit of electrical work, you're able to actually fuse the cells together, giving a little electric shock. The cells fused together, and what they created was the, oh my God, human-mouse hybrid. Notice that kind of the northern hemisphere of that has the human proteins. Southern hemisphere, so to speak, has the mouse proteins. Well, did it stay this way? No. Here's what happened after 40 minutes. Proteins are all mixed up. This is indicating that the proteins in the membranes are in a fluid medium and they move around kind of like icebergs in the ocean. So the proteins got all mixed up after 40 minutes. That was due to just typical diffusion of things. We have molecules that diffuse in gases, they do like the atmosphere. They diffuse in liquids. That's just from random collisions of molecules causing things to spread out and mingle. And that's what happened with these proteins. I've tried to find a picture of this and I haven't been able to, an actual photo of the cell. I would like to see what that looked like in a photo. Best I've been able to find is a diagram. But that's showing that the membrane is a fluid medium. Now, that fluid medium then is, part of the fluidity of it is maintained by the amount of cholesterol that goes in there. So cholesterol helps our cells to adjust the fluidity of the membranes. And if you have, say, an animal like a grizzly bear that's going into hibernation, its membranes are getting colder, and part of what the bear will do as it's getting ready to hibernate is it makes adjustments with the cholesterols. It makes adjustments with the fluidity so the membranes don't freeze up. If they just freeze up where they stop moving, that's not going to be healthy for the cells. Now, the proteins that are in the membranes can either be integral, where they pass all the way through the membrane, making something like a tunnel, or else they're just attached to one surface, peripheral. You can have a protein that just, say, attaches to the phospholipid heads, and the entire body of the protein is just on one side of the membrane. Or you can have it embedded. We get both types. Have receptors on them for a hormone, a signaling molecule. Hormone arrives from somewhere else in the body. Maybe it's insulin that is released because you just ate some pie and now you've got a rise in your blood sugar. Cells like in your liver or in your muscles will be signaled to start absorbing that insulin. So the hormone may arrive at a receptor, be received, and set off a reaction in the cell. The reaction could say, open up these transport proteins and let the glucose in, because glucose is otherwise trapped outside the cells. When you have a diabetic person, maybe type 1 diabetes, they're not making enough insulin because of damage to their pancreas. They will eat and they will get a lot of glucose in their bloodstream and it can't go anywhere. The cells have to actually open up, turn on these transport proteins to bring sugar in. Otherwise, the sugar stays trapped in the bloodstream, and that can be lethal if somebody has type 1 diabetes and doesn't get, say, insulin injections. But if they do get the insulin, then the receptor receives that signal and it tells the transporter, take in the blood sugar. You also have other transporters that use energy. ATP is a source of energy in cells, and they operate like pumps. Mostly today, we will be looking at these transport proteins, but there's other jobs too. We have proteins that will bind cells together. We have certain junctions, like the tight junctions between cells that prevent leakage between them. And so cells that are tightly junctioned together will have proteins from one cell that join right with a protein from another one. And we have these things called glycoproteins, and they present an ID for the cell, an identification, saying this cell belongs here, this cell should be in this body. You'll have a protein that has a small group of sugars attached. That's what a glycoprotein is. Glyco is referring to sugar and attached to a protein. Here we have the protein along with the sugar group, and then this other cell, a white blood cell, one of our defensive cells, is checking the sugars and making sure that cell should be there. If it doesn't have the right group of sugars, then the white blood cell will go on the warpath against the cell that doesn't have the right identification. And that's the basis of tissue rejection. If we have, say, somebody has an organ transplanted and their body rejects the organ, it's because their defensive cells don't like what they see right here. So we have these different roles that membrane proteins can take. Your book goes through a similar discussion. This figure's from a different book. And I will say again, none of this is on the test today. Do you have questions on these things before we go to the next part? Okay, again, we'll mostly be looking at the transport. So there's our question. How do various things get across membranes? Well, we have about 5 or 6 mechanisms, I guess 6 here to look at. One we call passive transport, and that is where substances simply zip right through the membrane, almost like it's not there. That happens with our oxygen. You inhale oxygen, it moves by passive transport into your blood. That means you don't need a special pump to move the oxygen, which is kind of a good thing. You know, there's less to go wrong with something that's a really critical function like that. Another thing is osmosis, which is basically like the first one, but it involves water. And water is such a dominant thing in us that when we get processes involving water, we tend to give those special names. We will see these first two pretty quickly. We also have facilitated diffusion. Here we're starting to talk about the transport proteins, moving things from one side of the membrane to the other. So you get these first 3 mechanisms. These all have one thing in common. They're not using energy. They're not using a power source. So your cells are not having to burn calories to make any of these happen. This next one, they do. This is where a lot of their energy goes, and that means a lot of your energy that you're burning in food calories and a lot of the oxygen you're inhaling goes to support this thing called active transport, which we will see. Then we have large-scale bulk movement of things into a cell, endocytosis. An extreme case there could be like one cell eating another one, swallowing it. Then exocytosis, which is basically the opposite. So we have the first 4, and these all involve moving, say, one molecule at a time. Endocytosis and exocytosis both involve moving a lot of stuff at once, bigger objects in or out. So when you have the phospholipids there, the bilayer, they have this rather thick nonpolar region with these fatty acid tails. Now, very small things like oxygen and CO2, those are small and nonpolar, which means they're easily able to zip through this. Because these phospholipids are in a fluid state, constantly shuffling and moving around, little gaps open up, and these small nonpolar molecules can zip right on through. I think of it being like with a screen, say a window screen. Imagine you've opened your windows, but you have a screen, and there's dust outside. Does dust make it through the screen?
Yes.
Sure. Very small particles come right through, and that's what these are doing. The O2 and the CO2, they're going through like dust particles through a screen. Now, the thing that's a little confusing here is that water molecules do it too. Even though we have this water-repelling double layer here, water still gets through. Water is about the same size as the other molecules, and molecules are colliding fast with each other. They're banging into each other. They're bouncing around. At body temperature, these molecules are moving pretty quickly. And as the phospholipids are constantly moving around, little spaces open up for a moment, and a water molecule is able to zip through one of those from one side to the other. In other words, membranes are leaky to water. When it comes to O2 and CO2, it's almost like the membrane's not even there. The membrane slows down water, but it doesn't stop it. So water is able to leak through here. And then we get other things that are either bigger or more polar. These ions have more of an extreme charge than, say, the oxygen in water does, or the hydrogens in water. These ions have full units of charge, and they are too polar to make it through the membrane. So if you have salt there and no salt there, the salt will stay on the salty side basically forever, unless you have a membrane transport protein move it. Other things like glucose that dissolve in water, amino acids, the various kinds of organic molecules we looked at, they are trapped on one side of the membrane or the other. They're either too big or too polar or both to get through the phospholipid bilayer. But we have membrane transport proteins that take care of that. They provide a tunnel and a special pathway that allows a substance to get from side A to side B. So, again, questions for me?
Yes, so what's on the left side here, the O2 and CO2 just passing straight through, is that passive transport?
It's passive transport.
Okay, thank you.
The other things that we see over at the right, those would all be facilitated diffusion or active transport. So, diffusion is just happening where molecules are moving around randomly, colliding. And if we have a membrane like this, imagine that the membrane has tiny openings in it. And initially, we have these small molecules, we'll say they're oxygen, and they're all banging around on one side. Now and then, one of them finds its way through. Look at the middle situation. They're going both directions. One of them is coming back right to left. But what's the predominant direction? Left to right or right to left?
Left to right?
Yeah, left to right. Because there's more of them over here, and there are more chances for one of these to move to the right than there is for one of these to find its way back to the left. The net movement here is left to right. It's going downhill. It is flowing from the side with the higher concentration to the side with the lower concentration. That is the spontaneous direction of diffusion, going downhill. Now, at equilibrium, we have an equal chance of one going either way, and so the concentrations stay the same. The first two situations, the flow was always downhill from high concentration to low concentration. That is what diffusion does. And this diffusion is happening because of body heat. It's basically just an outcome of the fact that heat is causing these molecules to rattle around and bang into each other. That's what heat energy is. And this is powered by body heat. It doesn't take a special power source to move things from left to right. All you need is a way for them to get there. So we have this downhill movement. We say the substance is diffusing down the gradient. The gradient is the difference in concentration between one side and the other. And the substance is moving down, spontaneously going from higher to lower concentration. And the steeper the gradient, the faster the substance will move. That means if there's a greater difference in the concentration between left and right, then there's going to be more rapid movement to essentially correct that. So that comes in when we're looking at things dissolved in water. We have here an osmosis situation. Imagine you have two compartments. One of them is just pure water on the left. On the right, we have something more like a cell. We have water molecules and, say, proteins in it. And then in between, we have a membrane. It's semi-permeable. That means, in this case, water goes through it, but not the proteins. The proteins are too big to fit through the membrane. So all the proteins stay cooped up on this one side. Given that, and the fact that water can move freely through the membrane, which way does the water go? Left to right or right to left?
Both.
It does go both, but on a net basis, which way is it going?
To the right?
Yes. The water is diffusing from its high to its low concentration. If it's 100% water on the left, it's less than that on the right, because part of that is the proteins. Maybe it's 80% water on the right. The water has to spontaneously flow downhill from its high to its low concentration. So that's what will happen. Remember that if you have more solutes, you have less water. And right now we're talking about water movement. Here's another demonstration of that. This is one you can do, and I haven't done it personally, but I've seen it done in a lab setting. You have a U-shaped pipe, and we put that semi-permeable membrane at the bottom with the little openings again that let water through. Here we have, in this case, the way I've done this before, seen it done, is to have sugar water. Here there's less sugar, on the right there's more sugar. So you have kind of dilute sugar water, strongly concentrated sugar water. Look what happens after a while. The water rises on the right. Did any of the sugar molecules go from right to left?
No?
No, they're too big, and if you count them, you see it's the same number in both situations. But what happened with the water? Did the water move anywhere?
Yes.
Yes. It went to the side with more solutes. And basically there's two things involved with that. You have these water-soluble solutes. Sugars and salts attract water molecules. You have here a solute molecule that is polar. It has binding spots on its surface where the water molecules want to stick to. And what we have is that we've got more water on the left, so there's a greater chance of it moving through the membrane, and then once it gets to the right, it doesn't want to come back. It's clinging to these sugar molecules. And so for two reasons, the water moves through here and ends up on the right side. The result is that the side that has more solutes attracts the water. And the way I think of that, real simple to remember here, is salt sucks. If you have salt on this side or sugar on this side, either way, Salt sucks, sugar sucks. Solutes suck. That's one way to think about it in more general terms. Anything where you've got more of these polar solutes draws water into it from areas that don't have as many polar solutes. That's what happens with osmosis. The membrane is leaky. Water does gradually get through it to the other side. So we get some terms that come out of that, all related to this idea of tonicity. That's a comparison of how concentrated the solutes are with two places separated by a membrane. And this is getting very jargony, and I'm sorry. But if one side is hypertonic compared to the other one, that means the side that is hypertonic has more solutes. Now, thinking about prefixes here, can you think of a medical condition that starts with hyper?
Hypertension.
Hypertension. What does that involve?
High blood pressure.
Yeah, high in other words. Hyper. Think about that in terms of something that's elevated, such as high blood pressure. Well, if the A side is hypertonic, it has more solutes. And that means if these two are separated by a membrane, water flows into the side that is hypertonic, into the A side. So the side with more solutes sucked water away from the one with less solutes. And a different way to look at it, if the A side is hypotonic, it has lower solutes. Can you think of another medical condition that starts with hypo?
Hypothermia.
Hypothermia, what's that involve?
Your body temperature got too low.
Yeah, it's low. So that's what hypo is referring to. So it's referring to the idea of something being lower than normal. So the side that has less solutes, that is hypotonic, gives up water to the other side, which then is hypertonic. This is always a comparison. It doesn't mean anything unless you're completing the comparison. There is a third choice, isotonic. Similar to isotope, isomer, we've seen this prefix before. And that means we've got equal concentrations of solutes and there's no net flow. Molecules of water are going back and forth, but they're not changing anything. So these are terms which I believe we will get into more with the labs this week, which I still need to post. Now, balancing water is crucial. Cells are mostly water. The animal cells, they have a problem if they're in environments that are not isotonic. Animal cells don't have a cell wall, and some of them are very flimsy, like red blood cells, and they will not keep the correct size or shape if you put them into something that is either a very salty solution or, say, a very unsalty situation, something like distilled water. So if you put an animal cell into, say, a hypotonic environment, say you put an animal cell into pure water, the cell will start absorbing water from the surroundings and eventually it could pop. That'll happen with red blood cells. They will definitely pop if you put them in that environment. If you take blood, put it into fresh water, all the cells just pop like little overinflated balloons. Now, if you have cell walls, that won't happen. Plant cells love it if you give them fresh water. So you go and water a plant with fresh water, its cells don't explode because the cell wall prevents that from happening. It reinforces the cells. So the correct environment for plant and animal cells is different. Plant cells want to be in fresh water. Animal cells want to be in slightly salty water. Neither one of them likes to be in extremely salty water, a hypertonic environment. That will cause all cells to shrivel. But has anybody been maybe visiting someone in a hospital and they've been hooked up to an IV? You ever see the label on the IV when it's just a drip of saline solution going into the person's arm? Well, if you're in that situation in the future, hopefully after COVID is taken care of, take a look at the label, and what you'll see is that the neutral saline solution is usually labeled 0.9% NaCl. That is slightly salty water. That is isotonic to human tissues, and it keeps our cells at the correct size. They don't shrivel, they don't burst. That's only about 1/4 as strong as seawater. Seawater is 3.5% salt. Our cells are about 1/4 as salty as that. But here's what happens if you have, say, animal cells, red blood cells. You put them in the isotonic solution, a little less than 1% salt, and the cells keep the correct size and shape. That's what a red blood cell should look like. Water moves in, water moves out at the same rate. The size of the cell doesn't change. You put it in very salty water, and most of the water leaves the cell, leaving behind a very shriveled, unhealthy cell. And we do this sort of demonstration in the class. So, sorry we're not doing it right now. Alternatively, if you put human red blood cells into pure water, this is what happens. They just explode. Plant cells, if you give them plenty of fresh water, their central vacuole fills up and it pushes the cytoplasm against the cell wall, and the cell wall restrains that, kind of like air pressure in a bike tire. You feel that, it's nice and firm, and that's basically what the plant cell becomes like. So then if you bite into a crisp celery stalk, it's because all the cells are essentially inflated like a bike tire. That's how they want to be. Alternatively, if you put the plant cells into slightly salty water, they will lose enough water that then they lose the Turgor pressure. They no longer have pressure pushing against the outer cell wall. The cells go limp. That's what flaccid means. And then if you put the plant cells into concentrated salt water, they shrivel. The plasma membrane pulls away from the cell wall. The cell is fatally damaged. So the optimal conditions for the animal cell are this one, and for the plant cell, this one. Do you have questions for me on this?
So is that how we get dehydrated if we were to drink salt water?
Well, that would sure do it. If you were floating in a life raft in the ocean and you made the deadly mistake of drinking seawater, if you were already dehydrated and you did that, it would just kill you. Seawater is poison for us. Here, 3 tomato plants, and on the left we've got the nice, happy one with the turgid cells. This was watered recently with fresh water. The cells look like we see down here. The one on the right, some stupid gardener decided to save on his water bill by using a bucket of seawater to water the plant. Not a good gardening technique. Here's what happened to the cells. Now this is something else. This is a Paramecium, and the Paramecium is a freshwater single-celled organism. It swims around in the freshwater, so it's surrounded by water that's about 100% pure. And it has the problem. Water is always leaking in through its membrane. Which is hypertonic here? The cell or the water around the cell?
The cell.
The cell. Maybe it has 80% water and the rest of it is going to be dissolve substances, ions and so on, that it's absorbed, proteins and various other things, glucose. The surroundings, well, that's as hypotonic as you can get if it's a lake. Inside the cell, it's definitely hypertonic. So which way is the water going to move? Into the cell or out of the cell?
Into the cell?
Into the cell. And this doesn't have a cell wall. What that means is the cell will swell up and explode. And yet, that doesn't happen. Well, let's see why here. See if I can find it for you here. Very short YouTube video, but this one's kind of nice. It even has some WC music in it. So you can see it happening. The only strange thing with this is that they keep freezing it right as the contractile vacuole squeezes. But nonetheless, it's a nice video. Watch this. So notice that the paramecium has two of these contractile vacuole organelles, and as water leaks into the cell, it squeezes it out every few seconds. That's part of the basic life of the paramecium. We have kidneys, they have contractile vacuoles. In a way, they're doing the same function. And if they stop doing this, they explode. So we had there our our passive transport, which is how things like oxygen and CO2 move between our blood and the air that's in our lungs, for instance. Those are always just diffusing downhill from high to low. Then water is doing the same thing, just more slowly. Those are diffusive processes. So is this one, facilitated diffusion. But now we have to have the transport proteins. So this is to move bigger stuff and more polar things. So you have, for instance, glucose needing to get into a cell. You have amino acids. Maybe you ate some food and you broke down proteins. Those amino acids now have to move into your cells so your cells can use them. These are all things that aren't able to go right through the lipid bilayer. So now we have proteins that have the job of moving one or maybe two of these substances. You need different proteins for different substances. So these are integral proteins that have a tunnel through them, and they allow you to move something from one side to the other. Different proteins move different things. At most, one of these proteins might move two substances, and we'll see a case of that. But otherwise, you have different genes in your DNA telling you to make all these different proteins to move all these different things. The proteins will change shape, then they change back. And these are not using a power source. The energy here is simply the energy of diffusion, which is coming from your body heat. So you have body heat, it puts molecules into motion, rattling around, and this is basically just providing those molecules a pathway to get from side A to side B of the membrane. As long as the flow is downhill, this is not requiring a power source. So the cells have to use energy to build these transport proteins, but then they're free to operate after that. So imagine you've got a particular solute like glucose. Trying to move it from one side to the other, it can't get through the lipid bilayer, so that's what this protein is for. Okay, I'm seeing a comment that somebody had their screen close up and then start back up. In general, are people's screens working?
Mine is.
Yeah.
Yeah.
Okay, at least one person had a problem. I hope it comes back to working, okay? Well, here, this is showing the glucose. Imagine you just ate a cake. Now you've got a lot of blood sugar. And your cells, let's say you're not diabetic, so they got the information from the pancreas telling them, take in this glucose. We don't want all this glucose out in the bloodstream. So the glucose transporters become active. And glucose can bind to a binding site, and then the transporter changes shape, releases the glucose, then it goes back to the original shape, and it can do this job again. Now here's a special instance of it involving water movement. We have these things called aquaporins, and these are doing special movement of water. We already said water can move through a membrane kind of slowly. What was the process for water doing that?
Osmosis.
Osmosis. It's not real fast. And because the membrane is putting up some resistance, it has that nonpolar region. Well, in some cases, we need the water to move a lot faster. That's in our kidneys. Your kidneys are squeezing a lot of blood plasma out into these tubules, and eventually that will go out as urine. And what needs to happen is that you get most of the water back, so you're not wasting it. We end up producing actually about an entire oil barrel's worth of this filtrate that's been squeezed out of the bloodstream. You make about an oil barrel's worth of this liquid, and you get almost all the water back in your kidneys. So you end up actually only urinating maybe a couple of liters during the day instead of hundreds of liters. Well, that's partly because of these aquaporins. The aquaporins allow very rapid recovery of water. And so we have these special proteins working in our kidneys, allowing the kidneys to get back most of the water. Otherwise, you would produce just a vast amount of urine, like hundreds of liters of it, and you would die. So these aquaporins work in our kidneys to allow rapid recovery of water, and they are actually facilitated diffusion proteins. So it's something like this. You have a special transport protein that's just working with water. You have water on the one side where you have fluid that is heading toward becoming urine. You want to get most of that water back into the bloodstream. And so some of the water is diffusing through the lipids by osmosis. But the kidneys have these special aquaporins that speed it up. So you have a whole series of binding sites inside the aquaporin, and the water molecules will stick to one, and then the next, and the next, and the next, and then they're ejected on the far side. One of these aquaporins can move about 1 billion water molecules per second, and that ends up being much faster than osmosis, which is having to drill right through the lipid bilayer. So that is a special case of a facilitated diffusion. Water is kind of special, and it actually gets 2 different mechanisms for moving it through a membrane. So, questions for me here so far? Okay. We'll get on through this, I think, fairly quickly here today. Let me check on the time. Okay, I think we're pretty good with the time here. So I'm going to bring us back to what we were just looking at. Let's see if we can get this done. So you have this other mechanism, active transport. This one's different. Everything we looked at so far involves downhill movement, things going from high to low concentration. And those don't, again, require a power source. Active transport does. This is pumping something uphill to go from low to high concentration. That takes a power source. And the power comes from this molecule ATP. We've just mentioned a couple of times. There's an organelle in your cell that is dedicated to making ATP. Which organelle was that?
Mitochondria?
Yep, that is their job, to take food molecules and take oxygen and produce lots and lots of ATP. And that powers the cells. And around half the energy in the cells goes into this process of active transport. So if you are eating, and say you're eating 2,000 calories in a day, 1,000 of those would be going to support this active transport. Or if you're inhaling 2 lungfuls of air, one of those lungs is supporting the active transport. So what happens with the ATP molecule is it contains 3 of these phosphate groups in a row, short chain of these phosphates. And there's a lot of energy in the connections between phosphates 1, 2, and 3. And what will happen is the ATP will interact with a protein and pass the 3rd phosphate to that protein, and it passes energy with it. The protein is energized, and now it does something. Changes the shape of the protein, changes what it's able to do, and energizes it to actually do something, pushing a substance uphill. So imagine you have this. We have a membrane. We have a high concentration of solutes on one side, low on the other side. The cell isn't satisfied with that. It wants to push even more of these ions to the other side. Well, they want to flow back. If they're just going to flow spontaneously, they would try to come back the other way. These down here are not going to spontaneously move in the uphill direction. But what happens is ATP will bind to this active transport protein. The binding of that phosphate energizes the protein. Its binding site changes, so now it has a high affinity. It's very likely to bind with this ion. And the ion binds, the protein changes shape, releases the ion to the other side, the phosphate drops away, and then the protein returns to its original shape. Another ATP molecule needs to bind with it for it to do this pumping action again. This is active transport. And this is where around half the energy in your body is going. We use it for a lot of things. For instance, inside your mitochondria, they rely on active transport as part of the whole process of making ATP in the first place. They involve concentrating hydrogen ions in one part of the mitochondria. We'll be seeing that a couple of chapters from now when we look at cellular respiration. Muscle cells, they require having calcium ions stored inside of some small compartments. And so when those ions then are suddenly released, it triggers the muscle to contract. But to get ready for the muscle to contract, you have to have these ions packed into these compartments inside the muscle cells. It takes energy to pack them in there. And your nerve cells involve a coordinated release of first sodium and later potassium ions. So you store sodium ions in one place, potassium ions in a different place, and then release them. First the sodiums and then the potassiums. And that's part of how a nerve impulse is sent. So these are different things we use active transport for. And here's the big one, the sodium-potassium pump. This is what's working in all of your cells, but especially in the nerve and the muscle cells. It's a complicated little thing. Let's step through this, though, piece by piece. You have a membrane, and what happens with all of our cell membranes, with the plasma membranes, is that they try to establish a high sodium concentration outside the membrane compared to the inside, and a high potassium concentration inside versus the outside. You have a pump that moves 3 sodiums out and then 2 potassiums in for every one of its cycles. So we have these pumps that are working all the time in our nerve cells, in our muscle cells. And first, what happens is the sodium-potassium pump binds to 3 sodium ions coming from inside the cell. ATP binds to this sodium-potassium pump and passes energy into the pump, and that causes the pump to change shape, causes these sodium binding sites to release the sodiums, and then causes these potassium binding sites to get ready to bind to a potassium instead. So with this pump, we use the ATP to push 3 sodiums out and then bind 2 potassiums, move them inside, and release them to the inside of the cell. So every ATP moves 5 ions, 3 out and 2 in. And this is the hungriest process in your body. We have so many of these sodium-potassium transporters that they account for at least a third of the energy usage in your body. So if you're looking at where your food energy is going, where the oxygen is going from your lungs, at least a third of it is going to operate these sodium-potassium pumps. This is the single hungriest process in the human body, requiring more energy and more oxygen than anything else. So we have the active transport, and it requires a power source. The way I think of it is like this: difference between passive transport, or let's say facilitated diffusion, and active transport. Imagine you're at school, and we're actually in face-to-face classes, and say you just took a test in biology, and you're really angry at your professor. You didn't think the test was going to be that hard. You were sure he wasn't going to ask you that question. And you want to get even, and you know his car is parked right outside. Is there an easy way to get even with him, just looking at his car? Maybe something involving his tires? No, everyone's afraid to say it. I'll say it for you. Poke a hole in one of his tires. What happens to the air when you poke the hole in there?
It leaks out.
Yeah, pretty fast. Did you have to pump it out?
No.
No, it just comes out on its own. When it's in the tire, it's at a high concentration compared to the atmosphere. All you had to do was give it a pathway, and it got out of the tire on its own. As long as you gave it a pathway, that's what the hole did. That's the idea with with facilitated diffusion. You don't have to pump anything out, it just moves. But active transport, let's say your professor wants to get the air back in the tire, what's he have to do?
You have to patch it first,
Yeah, that's the first step. Then what?
then pump it in.
Pumping, that takes work. Going back to having a high concentration inside, that takes work. That's where the ATP comes in. And if you were pumping it by hand, that's literally what you'd be doing, is burning the ATP to get the air back in the tire. So that's the difference with these transport proteins between diffusion from high to low versus pumping from low to high. Well, we have then these two ways of moving stuff in bulk: endocytosis and exocytosis. And here we're talking about moving a lot of stuff at once into a cell or out of a cell. Endo, you have some of the plasma membrane invaginates and it seals off into a bubble, a vesicle inside the cell, and it contains something that came in from the outside. So that could be, say, a food vacuole inside the cell. Exocytosis is the opposite. We have some of the plasma membrane buds off and pinches off into a bubble and escapes from the cell. It's releasing, say, cellular contents to the outside. So here we've got, for instance, endocytosis. You're bringing in some particles from outside, and now they are sealed up inside of a vesicle. Alternatively, you can have a vesicle fuse with the plasma membrane and release the contents to the outside. Phagocytosis is one way for this to happen. Here's where a cell actually eats something. So we have, for instance, a cell actually gobbling up, say, a food particle. That's what this amoeba is doing on the right. It's actually capturing some sort of a small organism. Now it's going to devour that. Once the small organism is sealed up inside of a food vacuole, then it can be digested with lysosomes. We have defensive cells, our white blood cells, phagocytes, and they actually will go and eat enemies. In this case, one of our defensive cells is trying to kill a whole parasitic worm by itself. That worm is a multi-celled animal. This single defensive cell is trying to kill that little animal. The defensive cell may die in the process, sacrifice itself. So we have these macrophages, and they go eating various enemies that get into us. And they'll either die in the process or else manage to digest the parasite. Alternatively, you get cells can simply drink some of the fluid outside, pinocytosis. Here, they're not being choosy. What they're doing is they're getting bigger. They're bulking up some by taking in some fluids from the outside, but it's not targeting anything. Here, it's actually targeting something. We have receptors that are clustered together in particular spots. You get these coated pits on cells, and they'll have receptors for something. So when the receptors get full, when they've actually bound to a target molecule that's out here floating by, then the entire collection of the receptors folds into the cell, bringing in these target molecules. That happens in us with cholesterol. Do we need cholesterol for our cells?
Yes?
Yeah. Where do we use it?
In the cell membrane?
Yes. Now, we actually make it in our liver. Even if you're eating, say, a vegan diet, which means, among other things, no cholesterol in your diet, you're still manufacturing it in the liver. And the liver sends it out through the blood in packages, and they arrive at cells. The cells can take the cholesterol particles inside and use those when they're making new membranes. Well, here's where that's happening. You have these cholesterol particles that have moved through the blood. They are binding to cholesterol receptors that are in one of these coated pits. And then here, that whole collection of the receptors, along with the cholesterol particles, is being taken into the cell. So that is a pretty efficient way of absorbing a lot of things that are one particular material the cell needs, and then it has them inside and it can use those as it's making new membranes. Alternatively, we release stuff, exocytosis. And so you may be making something using the rough ER and transport vesicles and the Golgi body. You may be making some finished material and finally release it from the cell. Well, that happens with making breast milk. Here we are in breast tissue, and we're producing the various substances that go into milk, things like the lactose and the proteins and the fat. Those are being then secreted into a mammary duct. Here, being released as one of these vesicles then fuses with the plasma membrane. So there we've got our different ways of moving things in membranes. You have questions for me on these things? In that case, I am going to give you something to do here for a bit. So let's go and take a look at my browser. I'm going to show you in here in, excuse me, in Canvas. Going to the home page, and as I scroll down here, I have a homework for you. This is due before midnight today. So it's 20 questions dealing with membranes, and they're all multiple choice questions. They're all related to what we just saw and to the second half of your chapter 4. Now you're doing the test today in class time, and then this assignment is something that would be due by midnight tonight, so you've got some more time to work on this. But I'm going to give you class time right now to work on this. So
which next to your screen?
Sorry, what?
Which one are you showing? Oh, there it is.
Yeah, membranes questions. So you'll find that under the week 3 module at the bottom of it in either, well, whichever Canvas you'd be looking at. It's in both of them. And so this is a set of 20 multiple choice questions, and you need to turn this into me in some fashion, however you want to do it. But you'll need to turn the answers in before midnight. Turn them in under the Assignments tab. So under Assignments, we will have down here under the Assignments, I have membranes questions. So that's worth 4 points, due before midnight tonight. So I'm going to give you some class time right now to work on this. And I want to break you up into groups, give you about 20 minutes in the groups to work on the questions. So I'm going to do that. Later, we're going to come back then and do your big test. So I'm going to break you up into 10 rooms here.